All right, thank you, Paul. So if you guys have any questions during my talk, just feel free to shout them out. You can try raising your hand, but there's a million bright lights in my face, so I probably won't see it. So it's best just to start talking. We can also try to take questions at the end, but it's better when you think of them. Here is a map of the psilocybin mushrooms that we have in the USA. And you can see that there's a lot on the West Coast here. They're a little more scattered on the East Coast, and then there's quite a few in the Southeast as well. And then also lots in southern Mexico, Central America. If we zoom into Georgia, we can see that Georgia has a lot of psilocybin mushrooms, most of them centered around the Atlanta area. And if you zoom in even more, you can see that most of these observations of psilocybin mushrooms are in the north, kind of like the northeastern part of Atlanta, from the city up over here, kind of, up to Auburn. But, this kind of part of the city that's like pretty close to Atlanta, but a little bit east and a little bit north, definitely a hot spot for psilocybin mushrooms. And the way I made this map is with iNaturalist. I excluded all of the observations that had obscured coordinates. So if you go out and you find psilocybin mushrooms and you found like 100 pounds of psilocybin mushrooms, and you don't want everybody to know the spot, you can set your coordinates to obscured. And that way people can only see to the nearest 10 miles where your spot is. So it's still really scientifically useful. If you zoom way out, you still a good point on the map. But if you zoom way in, you can't see. You don't have to do that. If I find just, a few pounds of psilocybin mushrooms, then I will leave the coordinates open. And that way anybody can see exactly where they were. If somebody wants to go photograph them, they can go photograph them there. And so this is a map of only the people who decided to leave their coordinates open so anybody can see exactly where they are. If you're interested in finding psilocybin mushrooms, I keep this list up to date, and this is on a website called the Shroomery. And this list has a list of all of the psilocybin mushrooms that are in every state of the country and in every country of the world. And so here you can see what we have for Georgia. We have quite a few different species here. And then if you go to this list, there's a link and you can click on that, see the evidence that this actually occurs in Georgia. But you don't have to try to read the URL on the top of there because I made a QR code. So you can just scan this QR code and it'll go to this list of which psilocybin mushrooms grow in your state. And so what you would do is go to this list and you'll see all these different species. Some of them are really rare, some of them are common. And so you can look all of these up on iNaturalist, Mushroom Observer, Google, Wikipedia. Just learn where in Georgia they grow, what time of year they fruit, what kind of habitat they like, what substrate they're growing on. And then if you go to the right spot at the right time of year, you'll definitely see some out there. The other day we found a bunch of morels and Psilocybe ceruleans. And there was like 1,000 Psilocybe ceruleans there, so they are really fruiting pretty well right now, even though it's kind of dry. They were super glue because of all of that. And it's... So the ovoids, they start first in the south. So they fruit first in Atlanta area, and then in a couple weeks, they'll be, in Tennessee. In a couple more weeks, they'll be in Pennsylvania and then Ohio. So they move north, and then so by the time June comes around, they will be all over. Like May and June is Ohio, northern edge of the range. Yeah. Sorry, did I say ceruleans? Oh, sorry. No, it was ovoids that we saw yesterday. If ovoids are now, ceruleans are later in the year. So I also made this URL, and it's this long URL that you can use to find psilocybin mushrooms on iNaturalist. And what I did is I put all of the genera and all of the species and then excluded the ones that don't have psilocybin mushrooms. And so I found that there was 533 observations of psilocybin mushrooms in Georgia, and with 17 species. So that is a lot of diversity of psilocybin mushrooms. He has more than most countries have, more than almost all United States except for maybe California. There is a lot of psilocybin mushrooms there, and the most common psilocybin mushroom here is Psilocybe cerulescens. So Psilocybe cerulescens was discovered in 1923 in Montgomery, Alabama. Turns out it is over there, but it's even more common here. And with Paul Stamets found it in 1996, he named it Psilocybe wileyi after his friend Andrew Weil. However, it's really the same thing that they discovered in Alabama. So I call them cerulescens here. But this Gymnopilus luteifolius is also very beautiful, bright red, purple scales on the top and bright orange gills. And then Psilocybe ovoids is the one that's fruiting right now. So those are really cool. They're very closely related to Psilocybe cubensis. So it's like a wood lover variety of cubensis. But you see we also have a lot of cubes in Georgia and Paneolus cyanescens. Yeah. So this URL, it's a pretty long URL, and I made it just so you can zoom in to like different parts of the world. You can, here I put in the state of Georgia, but you can put in like the whole world, or you can do just like a county or just a city. So if you want this URL, I made a QR code for it. So if you scan this QR code, then you can just zoom around the world. I think there's about 30,000 iNaturalist observations worldwide for psilocybin mushrooms. And about 500 in Georgia. So if you're like traveling to some other place, you can just put this URL, bookmark this URL, and then just go to it. You can see what kind of psilocybin mushrooms are in any part of the world. Yeah, so Gymnopilus dilepis is a name that is for a Gymnopilus that's really common. Or no, dilepis. Oh, that's like, okay, that one looks like luteifolius, but it's a little more purple. But yeah, that one is probably here, but it gets confused for luteifolius a lot. It probably is. There's one guy in iNaturalist that can tell them apart pretty well. Was it? Yeah, that's him. I've been seeing all his IDs on my notifications as well, and I think he's correct. I think he's figured out the difference. I can't really tell them apart, but they're okay. They're not deadly. Yeah, nothing in Gymnopilus is deadly. Most Gymnopilus species do not have psilocybin, but all of them with the red purple cap that's scaly, all of those have psilocybin. The dilepis and luteifolius definitely do. This is one that's pretty cool. Pluteus saupe is a relatively rare psilocybin mushroom. This one just turned up in Georgia, last year, but just a few days ago, the DNA sequence got back. So this lady, Ellen, who might be in the audience here, she collected this Pluteus and noticed that it smelled a lot like her psilocybin mushroom, smelled a lot like her ovoids, which are fruiting on the same log. And so she collected it, dried it, mailed it in to, I don't know where she mailed it, either the Ohio Mushroom DNA Lab or Stephen Russell. She mailed it to Scott. Okay. Well, so we got DNA sequence back for it, and that one is the first evidence that Pluteus saupe grows in Georgia. So that's another psilocybin mushroom to add to the list. But there's a bunch of different psychoactive Pluteus that grow in Georgia, probably at least 3. And it's kind of cool, they put some microscopy on here so you can see these are the cystidia here. They have horns on them. You can see the spores and all that kind of stuff. So there's a whole bunch of new things that we've discovered about psilocybin mushrooms in the last couple years, and one of them is that the blueing Conocybe just got a genus. So first we were calling them like Conocybe cyanopus, and then it was Pholiotena cyanopus, but now we actually have a genus for it. It's Conocibula. So here's a phylogenetic tree where you can see all the different species in the tree of life, and the ones that have psilocybin are right in here.
um, in these clades right in here. So Carnosebula is a new name for that. And some of the stuff in this tree are very toxic, some of them even deadly in Foliotina. But the ones in this group here, they all stain blue and have psilocybin. So this is what it looks like. A lot of these little brown mushrooms with orange gills, people don't usually eat them because a lot of them are deadly, like the Galerina marginata looks sort of similar. But these stain a very bright blue in the stem base. Yes, these can be cultivated, and they have been grown indoors. And they're not really that hard to cultivate. It's just like growing Psilocybe mexicana or something like that. And just do regular tech, and they fruit. Yeah, that's a good idea. And a couple of people have grown them, but the spore prints haven't really gotten out there. So somebody needs to grow them and then spore print every last one and just send out hundreds of spore prints of these things. That's a good idea. One thing about these is these are chemically different than all of the other psilocybin mushrooms in that they have real high levels of a tryptamine called aruginacin. So, psilocybin mushrooms have a lot of different tryptamines in them, but most of them are relatively similar in their chemistry, but this one is quite different. Also, another one that's really different is liberty caps. Liberty caps have a ton of baocystin, and so very, very different chemically from cubes, so they're likely to create a much different psychedelic effect than your standard cubensis. So in 2024 was a really good year for the discovery of new species of psilocybin mushrooms. There was 4 new species of psilocybe published in 2024. There was psilocybe niviotropicalis, Cerulea rhiza, maluti, and ingeli. So the psilocybe niviotropicalis was discovered by Scott Ostuni, who's right there in the purple shirt, and he discovered them in Jupiter, Florida. Congratulations, Scott. And so these ones are kind of closely related to Psilocybe ovoidiocystidiata, but they're much lighter color in the cap, and they're very picky about where they grow. They really like southeastern Florida, just right around here. So here is the paper that was published that gave Psilocybe niviotropicalis its scientific name. And most of the people that worked on this paper never went to college to learn biology or anything like that. So anybody can discover new species of mushrooms and name them. They do not check your credentials when you submit a scientific paper for publication. And here's some microscope work I did on it. This is like a cross-section of the cap. Here's what the gill edge looks like. And then if you crush the gill edge, you can break the cells free and see the whole cells. These are called chylocystidia. There are some spores, some pleurocystidia, cross-section of a gill. And then another new species that was discovered is Psilocybe Cerulea rhiza. And so this one grows in the Midwest, and this one I discovered in my mailbox. So it was like 2021, somebody mailed me some from Pennsylvania and also from Iowa City. And so I did a DNA sequencing run on them, and in the same batch of results, I got this new psilocybe barcode that hadn't been seen before, kind of close to Psilocybe aztecorum from Mexico and close to Psilocybe cerbica from Europe. But this one, here's where they turned up in Iowa City. And so these grow in wood chip landscaping, and sometimes there's thousands of them. Somebody found some in Michigan last year, and there was like 10,000 fruit bodies, just like right in front of City Hall. I think they're native to the Midwest, because we would notice if they were in other parts of the world, and they really have just turned up in the Midwest so far. And there's been quite a few finds. There's like 20 or 30 data points on iNaturalist. So they're not uncommon in the Midwest. A lot of different people have found them now. But yeah, I think they're native there. But the wood chips are like really concentrated food. But it'd be interesting to know where they actually evolved, because they certainly did not evolve in wood chip landscaping. Yeah, it probably is. Yeah, I met that lady in Michigan. She does a lot of collecting. Very, very prolific collector. Yeah. Yeah, she came to one of my talks in Michigan. This one's kind of interesting in that it turns up really late in the year. So you find it in November and December. So right when it gets really cold, this one fruits. And so I decided to name it Psilocybe cerulean rhiza because of the crazy rhizomorphs that you get on agar. And so this is an agar plate, and I just put a gill fragment in the middle of the plate, and after about 60 days it ended up looking like this. But this actually happens a lot. A lot of people have this culture. In fact, when I was down in Colombia in South America, I met this girl at a bar, and she had a whole purse full of petri dishes, and one of them was Psilocybe cerulean rhiza, and hers looked like this as well. This is this is the only psilocybin mushroom that has segmented cystidia. So you see these little septa and the cystidia. This is a really unusual microscopic feature. And then last year, 2 new species got described from Africa. So this is South Africa, and both of these were found by people who are not professional biologists or mycologists, just people who like mushrooms. So this one is Psilocybe maluti, and this just turned up on Facebook. This lady, Daniela Mulder, posted this photo in a South African Facebook group, and I'm like, wow, awesome, send them to me, I'll study them. And so she sent me some, and then Customs found them, and they confiscated them. And so then she sent the rest of them to my friend Brayton in South Africa, and he was able to study these and get a DNA sequence, and it actually turns out that they're really closely related to cubensis.
Are they illegal?
They are illegal, yeah.
Can you go back to the slide where you did the journal title?
The journal, yep. And you can see this one is an open access. So if you just Google this name, you can read this article without having to use Sci-Hub or LibGen to pirate it. So these are kind of cool in that they are secatoid. The caps never really open up. They stay closed, so they're almost like a puffball on a stick.
Wait, was that dung or?
Yeah, these grow on cow dung. Yeah. And then the other one is Psilocybe ingeli. And Psilocybe ingeli, also found in South Africa, but it was found out in the grass. And so this one is really closely related to Psilocybe zapotecorum. So Psilocybe maluti has proven resistant to cultivation attempts, but Psilocybe ingeli is really easy to cultivate, and it's extremely potent. Somebody just gave me some spores of this the other day, but this would be a really good one to cultivate. It can be 4 or 5 times more potent than Psilocybe cubensis. So in 2025, it might be another really good year for the discovery of psilocybin mushrooms. There's a couple of them that I'm working on that I find out west. And these grow in the mountains of California, like really high elevation around Mount Shasta, and really beautiful mountain meadow habitats. And then in New Zealand, there's a lot of psilocybes that have not been named yet. And we also found some in Ecuador where we sequenced the DNA barcode and they don't match anything. So lots from there. And then there's one, this psilocybe from the Pacific Northwest. This one I got a good collection of in Arizona as well. And then there's one that turned up in California, in Southern California, and several new Paneolis.
Explain just real
Yeah, so this psilocybe species CA03 is a provisional species name. And so if we get a DNA sequence back and it doesn't match anything that's known, then we can just give it a provisional name. And we could give it a fancy provisional name, you know, you can call it whatever. But the easiest thing to do is just call it like California 3. So that means there's two other ones also in California.
Same species.
No, the other ones are different. So Psilocybe California 01 is different. Psilocybe California 2 is different. All three of them are probably new species.
Yeah, the one, the number is what gets me. Okay, so it's
Yeah, so that's just randomly assigned. Like if I am processing DNA sequence data and I can't figure out what it is, I'll just assign one of these. And if there's already 10, I'll make it 11. So this is the one that we're going to call Psilocybe gandolfiana. And this one's pretty closely related to Psilocybe hopii, which was discovered in Arizona. And this is another one that's like a puffball on a stick. Pretty potent one. It's kind of a wood lover. And then Psilocybe polytryptamine. How do they spore out? That's a really good question. And it's kind of hard to tell, but a lot of these are in meadows, but kind of near where the water would run in meadows. So it's possible that the water spreads the spores quite a bit. Also, maybe birds will like spread them around. But yeah, they certainly are not going to be flying around on the wind like a lot of spores will.
Would that also apply to Baluti?
Yeah, for sure.
So,
Yeah.
what?
let me throw in, I was asking if that also applied to the Baluti.
The Psilocybe maluti, the South African one, is also like this.
And then let me piggyback on that question. So it seems to me like there was something special about exporting the Baluti because you've got natalensis, real natalensis, Braciocentrata, but there seemed to be an extra barrier. Does that have to do with the kingdom of Lesotho in Africa? Any special reason?
It's really just the way they were sent. So if you send spores in just a regular letter, then they generally make it, even to like some restrictive regime somewhere. But the ones that were sent to me were sent in this big package with like special courier service, and they generally open those. So if you want your spores to arrive, you know, you just put them in a normal letter with a normal stamp, and then it just goes along with every other letter. And they don't really open letters, but they do open packages, and this was more of a package.
So you're saying that no one has tried, I'm sorry, but just talking to some people, like, as far as, it has nothing to do with the kingdom of Lesotho being in the South African region.
That's one of the places that they found it. They found it in the Kingdom of Lesotho. But I don't think those are the people that took the mushrooms out. I think it was the South African government that did that.
Question for these mushroom structure. Take a cross section. Actually, two questions. Did you do the cross section of the mushroom? I'm very interested to see what's inside of the cap. I don't know how to find that. And if you cut it open, you see geostructure. What is it?
Yeah, I did do cross sections. I always carry brand new razor blades in my wallet, and that's really good because then whenever I'm out in the field, I have them. Even if I fly in the airplane, they don't see them. So brand new razor blades will make a much better cross section than like a pocket knife will because it's so much thinner. I think I might even have, I don't know if I have a cross section here. Looks like no, but I have them on iNaturalist and Mushroom Observer. But this one here is another one of these secatoid mushrooms, so they never open up. And this one is really closely related to Psilocybe pelliculosa. So these secatoid mushrooms seem to be an adaptation for dry areas. So places like California or South Africa that don't get much rain, the mushrooms adapt by the caps never open up, and then they're just more like puffballs on sticks. But this one is also found at really high elevation in California. And you can see it bluing pretty well on the stem base here. So this one will be Psilocybe polytrichoides. And I should add a cross-section to this because I did take a lot of really nice cross-section photos of this spring. And then here's Psilocybe PNW02. So this is another one of those temporary codes. And where I find it is right in the White Mountains of Arizona. It's a really beautiful place. You can see it's one of the very green parts of Arizona. So this is one of the best places in the world to hunt mushrooms. The moisture comes in off the Gulf of Mexico in August and September, and the amount of mushrooms in those Arizona mountains is just ridiculous. But the same thing has turned up in Idaho and also a few times in Washington. So this is what PNWO2 looks like. It's closely related to Psilocybe folliculosa. And then Psilocybe aztecorum is another really cool species, and this was discovered in Mexico, a place called Popocatépetl, which is an active volcano right near Mexico City. So if you've ever flown into Mexico City and you see that snow-capped volcano right there, that's Popocatépetl, and that's where this was discovered. But just this year, it was found in Oregon by Jordan Jacobs, and then in California as well. So this is a really high elevation species of Psilocybe, and it has these cool rhizomorphs on the stem base, and it really likes cold weather. It's also turned up in Colorado. And then here's Psilocybe species California 3. And this one was found by Andrew Kunick in Southern California. He might be in the room now or maybe not. But this one is close to Psilocybe ovoidiocystidiata, and it's likely that the spot that this was discovered burned in those Los Angeles wildfires. And then here's one of the New Zealand ones. This is a species of Psilocybe that's close to Psilocybe stuntzii. So you can see the annulus here. But this one I found at the New Zealand alternative to Home Depot. So it's a really big box store. This is what it looks like in ultraviolet light, and this is in white light. But in New Zealand, we actually go to these big store, it's like a massive hardware store with a garden section. And we go to these big stores to look for psilocybin mushrooms. And you don't really find very many in this country, but in New Zealand, if you go to the garden section, there's mushrooms coming up in a lot of the pots. And so we figured that it was going to be like a quick raid while we run in there, photograph the mushrooms, get kicked out in 3 minutes. But actually, the employees, they like saw us, setting up the black velvet and the flashes and the tripods and everything, but they didn't say anything to me. And we spent like half an hour with these mushrooms photographing them, and it was really nice. They're just really polite there, I guess. Psilocybe mushrooms are illegal in almost every country, including New Zealand.
I'm sorry, I'm trying to, you mentioned wildfires a few slides back. And I know that you discovered some morels here in DeKalb County. We haven't had any fires. Can you just tell us, specifically what type thrive after wildfires and which ones you found in DeKalb County?
Yeah, so there's basically 3 kinds of morels. One kind is called a landscape morel. That one grows in wood chip landscaping, and it grows all year round whenever it rains. I don't think you have those around here. The other kinds are called natural morels and burn morels. So burn morels are very common in the western United States and western Canada, but they're pretty rare in the east. We do get them here. Sometimes there's fires like North Carolina, and there'll be a bunch of morels. But for the most part, 99% of the morels that we find in the eastern half of the United States are naturals. So they don't need a fire. They don't need a tree to die or anything. They just fruit naturally with no burn. And that's what I found this Friday.
And that's what we call wood chip morel?
No, the wood chip ones are more of like a West Coast thing. So I don't think you get wood chip morels out here. They're just natural morels. And so they grow a lot with ash and elm. The guy that, tulip poplar, but I think it's a different species with tulip poplar. It might be like Morcella diminutiva. But yeah, tulip poplar is a really good morel tree in Georgia. But the guy that found the spot that we were photographing the other day, he says that he looks for box elder. but I think it might actually be growing with ash because the ash kind of grows near box elder and there was some ash in the area. But the advice that he gave me is if you want to find morels in Georgia, almost all the good morel spots, you can hear a train from where the morels are. And that's because the morels like these flat areas that are near creeks, and the train tracks also run in these flat areas near creeks. So he says all of his best morel spots, there's a train within earshot. But the biggest thing to look for is ash trees. So if you can learn to identify ash trees when they don't have their leaves, that would be a good thing to do. And the bark of an ash is pretty distinctive. A lot of people teach tree identification classes in the winter, so you can figure out the trees.
The the ovoid, I can't remember. I find that they're around box elder as well.
Yeah, so ovoids do like box elder. They're not super picky about the wood that they consume, as long as it's not conifer. I don't think they like pines very much, but they'll definitely consume box elder for sure. But a lot of they like oak, pecan, a lot of different hardwoods they like. And so both morels and ovoids fruit very early in the season, and so they're both fruiting right now. And in a month, they'll be done. And so, morels only fruit for about 3 weeks in any given location. So,
Found all of them so far in Georgia.
what's that?
I think you found all the morels in Georgia.
No, I only spent 10 minutes looking for morels in Georgia, and I picked 10 of them in 10 minutes, but I only checked one spot. I think there's a lot to be found. What's that? Privet. Yeah, somebody else told me that they really like privet too, but privet's invasive from China, so I'm not sure if the morels like privet or the morels just like the same places that the privet likes. But there's definitely a correlation, yeah. So another one of these New Zealand ones that hasn't been named yet is Psilocybe subsicodioides. And this one is super potent and it grows in wood chips and it's kind of halfway between a puffball and a mushroom where the cap opens up. So here you can see the cap opening up a little bit, but it's also kind of puffball-like. And then this is the one that's completely puffball-like. This one is also in New Zealand and Australia, and this likes to grow on tree ferns, but it's a psilocybe. And here you can see the cross-section. The gills are a little bit wrinkled in here, but they're kind of cool, and they have psychedelic puffballs in New Zealand. And then here's Pluteus phaeosanapis. It's a rare Pluteus. This particular one is on the west coast, but the features are the same everywhere. You can see that these mushrooms have free gill attachment, so the gills do not touch the stem. And so you got the stem going up, and then the gills go in, and there's this ring around here. All the different Pluteus do that. So you probably have at least 3 different psilocybin-containing Pluteus in Georgia, and they all have free gill attachment. And they also have pink spores. So the gills start out white, and then the gills turn color to pink as the spores mature. And paying attention to how the gills change color as the spores mature is a really important thing to do, because then you can see the spore print color without having to actually make a spore print. The Pluteus are not super potent. They're usually a strong one is 1% psilocybin, so that is about the same as a regular cube that you would find in the fields out here. Another really interesting one is Inocybe. And so most Inocybes are poisonous. They have a toxin called muscarine. But this one, Inocybe insignis, turns out it stains blue and it has psilocybin and it grows with oaks. So you might find this around here. I tried a Miraculix test on this. So Miraculix is a home test kit that was invented in Germany. And it takes about 15 minutes, and then you can test the potency of psilocybin mushrooms. And it seems to work pretty well. It's accurate to within about 10%. So you can see this, this inocebi, this one came in the mail from Ohio, but it is about 2% psilocybin, so strong, about twice as strong as an average cubensis. And these things are absolutely beautiful in the microscope. This is called dark field microscopy, so you get a jet black background. And here's what the spores look like. And then another really interesting one is this tuberia. And this tuberia also turned up in my mailbox. Most tuberias are really boring and don't stain blue, but this one was staining blue. So he mailed it to me and I sequenced it, and it turned out to have a novel DNA sequence. It's now turned up a couple more places, but this one is about 1% psilocybin, so not quite as strong, but... What's that?
From the US?
Yeah, that came in the mail from Pennsylvania, and then it also turned up in Ohio. So it's probably all over the Midwest. And then this one is not a new species, but just kind of a cool picture, and I just took this picture recently when I was in Colombia. So this one is Psilocybe hoogtjegenii, and these just have a really acute umbo on the top of the cap. And if you read the literature says that these are like kind of closer related to Neohalopensis and the Cordyspore. But I brought some back from Mexico and I sequenced the DNA, and it turns out that it's actually really close to Psilocybe mexicana. And so it's just a really unique psilocybin mushroom.
Is that growing on bamboo?
This one here, it kind of likes grassy areas, and it also likes landslides.
Bamboo's grass?
Yeah, bamboo is a grass, but this is usually like smaller grass. I think it would grow on bamboo, but you find it like out in fields, and yeah, also just like if there's a massive landslide, you'll find it in there too. And there's a lot of cool new papers that came out recently on the chemistry of psilocybin. Turns out most of the papers that come out about psilocybin chemistry come out from this tiny town in Germany called Jena. And it's this little town in the middle of Germany, but they've discovered this chemical norpsilocin, and they've actually discovered almost a dozen different tryptamines now in psilocybin mushrooms. So when you have psilocybin, you always have psilocin, aruginacin, baeocystin, but different species have different ratios of these tryptamines, so that probably is the reason that different kinds of mushrooms will cause different effects. And also, even more recently, they've discovered that there are beta-carbolines in psilocybin mushrooms. So beta-carbolines are in the ayahuasca vine, and they are the chemical that gives the ayahuasca vine its MAO inhibiting properties. And these same beta-carbolines have turned up in psilocybe. It turns out there's not very many in the fruit bodies of Psilocybe cubensis, but the mycelium of Psilocybe cubensis has a lot of beta-carbolines. And also the sclerotia, the philosopher's stones, Psilocybe tamponensis, that has quite a bit of beta-carbolines too. So in the Atlanta area, they discovered Psilocybe tamponensis, but they thought it was a new species, so they called it Psilocybe atlantis. But it turns out that Psilocybe tamponensis is the correct name for Psilocybe atlantis.
Next section, Mexicana.
Yeah, really closely related to Mexicana, but different.
So could they, if you were, you had a single spore of each,
Do they cross? Do you know if tamponensis crosses with Mexicana, Scott?
yeah. What?
Yeah, they're pretty close. They're about 6 base pairs different in the ITS gene, so that's close enough that it would not surprise me if they could cross. Yeah, that's a really good question. Like, are there any correlations between what they're growing on and the chemistry? And yeah, Jordan Jacobs has a psilocybin testing laboratory in Portland, and he finds there's a little spike on his graph that's present in wood lovers. It's not present in anything else. And so, you know, it's probably not the wood itself doing that, but just they have different genes that produce psilocybin in the wood lovers, and probably the reason to get the wood lovers paralysis and everything too. But yeah, there's definitely a different chemistry in there. And then my friend Marshall in Jamaica, he was growing a whole lot of mushrooms on core and vermiculite, and then also on dung, and testing them with HPLC. And he found that when he grew mushrooms on dung, they were about 1/3 more potent than they were with core and vermiculite. So that definitely works to some degree, though there's some people that claim to discover like all these other compounds like psilomethoxin in mushrooms, and that's actually just complete nonsense. So you can't just put like 5-MeO-DMT into the substrate and have it 4-phosphorylate it and make other psychedelic drugs. There is peer-reviewed research out there that says that you can, and that peer-reviewed research is wrong. But it's never been replicated. You know, they published it 40 years ago, and nobody's ever been able to do it again. So. A lot of these mushrooms have enzymes that are breaking down the lignin in the wood, and so, you know, it's really complicated. There's hundreds of chemicals in wood, but the main structural thing is the lignin, and they're definitely breaking that down. This research here is kind of cool. This is called MALDI mass spec, and MALDI mass spec, what you can do is it's kind of like a microscope, except that the microscope can see different chemicals. So this is a cross-section of psilocybe mycelium, and you can see where in the mycelium the beta-carbolines are, and you can see it's all in this outside part of the mycelium. So like the inner core of the mycelium has almost no beta-carbolines, and the outside has a whole lot. So this is a pretty cool technique because you can see like what parts of the mushroom are most potent or which cells are creating psilocybin. And then even more recently, this same lab in Germany discovered that that magic mushrooms also have terpenes in them. So a lot of people know that the terpenes in cannabis affect the the feeling you get from smoking weed, but terpenes are found in mushrooms too, and the research on this is just starting. But a new paper is going to come out in a couple months about this. But this terpene here, they named it after Psilocybe cubensis. And then some of these terpenes are the same ones that they find in cannabis. But there's a whole bunch of different terpenes in here. And terpenes, yeah. And so people don't really know what effect this has on the trip, but it probably does have some effect on the psychoactive, part of the mushroom. So it turns out if you put a mushroom into a mass spec, you'll see there's like 1,000 different natural chemicals present in the mushroom. And when you eat a mushroom, all 1,000 of those chemicals are interacting with your body to create the psychoactive effect. And most of them don't have any effect, but if you have 1,000 chemicals, some of them will, and they'll all interact to give you that experience. So the chemistry of these things is very complicated, and it's not understood yet which chemicals are doing what. Another thing we've been learning a lot is just making phylogenetic trees with the DNA barcoding. And so here we have a phylogenetic tree, and this is from the paper that describes Psilocybe maluti. And you can see this is the cubensis group over here. And so what we can do is sequence all these mushrooms and figure out how they all fit together, and kind of see like where in the tree of life each species fits in. And this is something that we've only recently been able to do, and it's something that's pretty easy to do at home. So you could set up like a DNA sequencing lab at home for under $1,000 and be discovering new species and building these phylogenetic trees. Or you can just mail the mushrooms into larger labs that do all of this.
Are you saying to buy a PCR?
Yeah, all you really need is a PCR machine and some pipettes, and maybe a gel box, but yeah, you don't need too much. So the numbers on the left is called bootstrap support, and what this is that when you build a phylogenetic tree, the computer tries to calculate how likely it is that it is correct, that it put everything in the right spot. So a higher number means that the computer is more confident that it's correct, that it actually was able to figure out how these things are related. If you have a real low number, then the computer is just guessing, and it's just going to put these things in a random spot in the tree because it doesn't really know. So higher numbers are better, and you can get higher numbers by sequencing more genes or having different things included in your tree. But like this tree here, the numbers are pretty high, so the computer probably did figure out how these all evolved. This section here is really interesting. The Zapatocorum section has a lot of the most mysterious species, a lot of the most potent species, and there's only one in the United States in this section, and it's found in New Jersey.
At a glance, I don't think, oh, okay, I see it way down there. I was looking for subtropicalis.
Yeah, so Subtropicalis is extremely closely related to Psilocybe zapatocorum. The DNA sequence is only one base pair different. So out of 600, there's only one difference. But it's really easy to tell apart in the field. They're very clearly different mushrooms. So that one's kind of interesting.
You know, it's interesting. I did the Cal Farmers Market, the Grand Park Farmers Market earlier today, and I see the vendors are catching up with summer enoki. It's familiar with the summer enoki take, which is very different from the white. It really looks, you know, one of us might walk up and think it was Subtropicalis. Have you looked at that?
I haven't seen those, but there's definitely a bunch of different Flammulinus that they grow.
Yeah, if you take a look at the summer one, the winter, so the winter enoki is grown in darkness, like as the, what do we call it, common button mushroom, which they kind of force.
The white ones.
The white, yeah, and so we always see enoki as a white mushroom, and the summer enoki, to me, I think I have a picture on my phone, but it looks really like you could, to me,
That's cool. I'll have to look for those. And yeah, when I see those things in nature, they have quite a bit of color to them.
it looks like Subtropicalis.
But one thing I would like to encourage people to do is grow less cubensis and grow more of these other species, because cubensis are kind of a boring mushroom. They're not super potent, and everybody grows cubensis, and it is very easy to grow and you get a lot of weight. But I think a lot of these other species, like Campanensis, Zapatocorum, really any of these other ones are much more interesting because they have different psilocybin genes. They'll have more different, more varied chemistry than cubensis. Also, like cubensis kind of hurts my stomach, but if I eat like a bunch of Zapatocorum, I can eat a whole lot. It does not hurt my stomach at all. And so I think I really hope that people don't stop growing cubensis, but stop growing so much cubensis, and grow a whole lot of these other more interesting species. This group over here is a whole bunch of very mysterious little brown psilocybes that you find in Central and South America. And then this is a completely different kind of fungus. This is a map of where you find Claviceps paspali. And this is not just a map of Claviceps paspali. This is a map of where in the United States there's people that live that know how to identify Claviceps paspali, because it's really everywhere. It's just hard to identify. But this is a photo that I took of Claviceps paspali. And the cool thing about Claviceps paspali is that it makes ergot alkaloids. So most people think that LSD is produced from the rye ergot, Claviceps purpurea. It turns out that it's not really true. Almost all of the LSD that's produced these days is produced from Claviceps paspali because it's much easier to cultivate. So this grass here is called paspalum, and paspalum grass is very common. And if you look real close at paspalum grass, you might see that it has a sticky top. And if you have paspalum grass that's sticky on the top, that's this fungus, Claviceps paspali. And what it's doing is attracting the ants and the bees, and this sticky liquid is full of spores. But it also makes sclerotia. So this is a sclerotium of Claviceps paspali. And if you take this, cut it open, throw it on agar, you can fire up a culture. Here's the kind of sticky liquid. Another thing that's kind of cool and also kind of mysterious is the ayahuasca plants. This one here is a plant called Diplopterus, and this is the main plant that they use to make ayahuasca out of in South America. But the identity of this plant is kind of in question. The book says Diplopterus cabarena, but the flowers look totally different. So it's nobody really knows what this plant is, but it's used by the native people all over South America. And it may or may not have a name, but it's absolutely beautiful, and it has really high levels of DMT. So that's the DMT part of the ayahuasca drink. And then here's a paper that I published with some friends a couple years ago. This one is about Psilocybe zapotecorum. And Psilocybe zapotecorum is one of the most potent psilocybin mushrooms. And so this paper has really detailed cultivation instructions, so you can grow Psilocybe zapotecorum indoors. And then we also did a full genome sequence on it and took apart the psilocybin gene cluster to try to figure out why it's so potent. And so really cool mushroom. These things can grow like a foot tall. And yeah. Yeah, so this paper is open access. So if you Google like the title of this, you can find the PDF online, and it's a pretty cool paper. But this is my favorite mushroom to photograph because it's extremely potent. It stains blue like crazy, and lights landslides. So you find it like in disturbed areas, and it's a really cool mushroom, and it smells like cucumber and radish. And yeah, it stains blue really good. But I think this is my favorite one to eat. Like if I eat a bunch of this, it feels like a 5-hour DMT trip. And it doesn't. If I ate enough cubensis to do that, it would definitely get a stomach ache. So I think more people should grow the Psilocybe zapotecorum, and I think it would do really well outdoors in Georgia. It would probably take off just like the saberlessins and the ovoids have. Also, if you're collecting psilocybin mushrooms, it's a really good idea to not only collect the psilocybin mushrooms, but also pick all of the other mushrooms that you see. So here we have Paneola cyanescens, but I also pick all this other stuff. That way, if you're stopped by law enforcement, you can just say you're picking all of the mushrooms instead of just the psilocybin mushrooms. Yeah, that's Marasmius vagus. Yeah, this is one of my boxes from Michoacan. First time I saw it in Mexico was like 2012, but I wouldn't be surprised if it's been there for a very long time, but I also wouldn't be surprised if it's only been there a few years. No, not psychoactive, but it's beautiful. I'll try to look it up real quick.
How would it come over from Australia?
I have 46 images here, and if you look on the right, it's going to combine those 46 images into one image, which has more depth of field. So if you look on the right, you can see the image starting to come together. And you'll see a little bit more. But this focus stacking technique, it allows me to have infinite depth of field, so as much depth of field as I want, and also leave the background very blurry. So it's a technique that's kind of revolutionized photography. And I was never really happy with how all my photos turned out until I started focus stacking, and now the photos, it seems like just like every one turns out just how I want it to.
Do you use a manual slider for like
So this particular one's not with a manual slider. This is with a Nikon camera, the Nikon Z, and they can focus shift shooting. So it just takes a lot of pictures real quick and changes the focus. But see, if we zoom in here, we can see that there's really good detail, and everything is just like razor sharp. This one is Psilocybe allenii from San Francisco. No, not only Nikon has it. Olympus has had it longer than anybody. So if you want to get a used camera that has that feature, the Olympus ones are the least expensive. Canon has it now in the high-end cameras, and then only the brand new Sonys have it. But it's a really good feature. So if you're looking to buy a new camera for mushrooms, definitely make sure it has the focus bracketing feature. And you can message me, and I'm happy to give out camera advice. And yeah, so if you pick mushrooms, always pick all of the mushrooms you see, not just the psilocybin mushrooms. So if you run into law enforcement, you can just say you didn't know what any of them were, and you were just picking every mushroom you saw. And also, you can take all of these mushrooms and send them in for DNA barcoding. There's only 246 DNA barcoded mushrooms from Georgia on iNaturalist, and we really need more. For example, in California, we have about 25,000 DNA barcoded mushrooms. So what we need is people that pick mushrooms in Georgia, take good pictures, and save dried collections and send them in for DNA barcoding. So if you want to help the mycology in Georgia, that's something that everybody can do, is start collecting dried mushrooms. Not my mailbox, unless it's like a psilocybin mushroom or something. But if you let me know, we can find somewhere that you can send them. But yeah, definitely take the pictures, put them on iNaturalist, save the dried samples, and label the dried samples with the iNaturalist observation numbers. Thank you very much.